Okay, shall we start, sir?
Yes, please.
Okay, thank you, sir. Good afternoon, all. Myself, Dr. Chintan Somaiya, Assistant Professor, Department of Chemistry, Parul Institute of Applied Sciences, Parul University. First of all, welcome to all to join this webinar on analysis of natural products in plants. I also thank to Dr. Azaz Ahmad Qureshi, sir, for accepting our invitation as an expert for today's webinar. Sir has completed his PhD from SVNIT, Surat, in collaboration with ICAR. During this, Sir has published many research papers in national and international journals. Also, Sir has published many book chapters in reputed journals. He has also participated in many national and international conferences. He is currently working as a scientist in analytical development and innovation sector at Farmanza Herbal Private Limited. Okay, so thanks, Dr. Azaz Ahmad, sir, once again. Now, I request Dr. Azaz Ahmad Qureshi, sir, to begin your session. Sir, please.
Thank you so much, Dr. Chintan, for this wonderful introduction. I would like to start my presentation with my own PhD topics photograph. These fruits are Garcinia xanthochymus, which is found in the forest of Assam, and this picture was taken by me in the year of 2017. So today's topic is based on analysis of natural product in plants, and it's very wide topic because natural product itself says the products which are originated from the nature, and it can be classified further from animal based, microorganism based, or plant based. So I have focused only plant based natural products and its analysis. The presentation includes the introductory part of this particular natural products and importance of plant based in our day-to-day life, as well as further extraction will be discussed in depth, and some of the extraction techniques from conventional and non-conventional will be discussed, and after that qualitative and quantitative methods for the analysis of plants and their products will be discussed. Note that I have given you acknowledgement to the images and other contents to their references. So as per terminology, natural products is nothing but products of natural origins, and it can be classified in major three origins. One is animal, and then microorganisms, then plants. One example is vitamin A, also vitamin D, which is obtained from cod liver, which is animal based. Here is the chemical structure of an vitamin A. Then another one is doxorubicin. It's a well-known anti-cancer drug which is obtained from a bacteria, and this complex beautiful structure, if we synthesize in lab, it takes a lot of patience, lot of efforts. And the other one, paclitaxel, which is also known as Taxol, which is obtained from Taxus brevifolia plant. So let's move on to the classification and what are phytochemicals. we can say phytochemicals as phytochemicals also, because these are the chemicals which are helpful to fight against various ailments, various diseases, and even for cure of various illnesses. And basically phyto, it is originated from phyto in Greek language, which means plant. So these chemicals are known phytochemicals, and this particular chemistry related to this topic is known as phytochemistry. And these are the major classes of phytochemicals: polyphenols, carotenoids, alkaloids. You might have heard about these things in your UG or PG. I have kept this presentation as much as simple because UG and undergraduate or postgraduate student can uh, grasp easily. And to know about this particular underrated topic, I am calling it underrated because we all of in our postgraduate or undergraduate, we don't know how this topic is. It is a very beautiful topic, but we are not aware with its basic things. Whatever our ancestors told, that tulsi or what Adha Toda, we call it Ardusi, these are the day-to-day routine plants which we are using since our ancestors have told us about its benefits related to different kind of coughs, or we can say cough, cold, and etc. So these are made up of various compounds, and which can be classified into different phytochemical groups. So what are medicinal plants? Medicinal plants are plants, herbs, shrubs, climbers, which can be taken as medicinal purposes, are medicinal plants. And it has been estimated that more than 250,000 plants have been identified throughout the world, and out of 250,000 there have been 70,000 only characterized for medicinal purposes. So one can say there are so much scope in upcoming future, so we can understand the topic and its basic there, so that we can further explore this beautiful topic, and we can do research and find out new drugs for the various diseases. Out of these 250,000, approximately 18,000 plants have been identified in India, and India is known as Botanical Garden of Asia. So this is just a stat. We cannot say whether it is 18,000 or more, but out of 18,000, only 6 to 7,000 medicinal plants have been characterized for medicinal properties. And till date, greater than 75% of population still rely on plant-based medicines. So when you have cough or cold or fever, your grandfather or grandmother will suggest you to take tulsi, ardusi, what we call Adha Toda in English. We are habitual to take those things after heating or boiling in water. And 30 to 50% of drugs and nutraceuticals are plant-based. More than 60% of anti-cancer drugs have been originated from plants, directly or indirectly. And if we talk about the last 4 decades, there have been 929 drugs which are newly approved. Out of nearly 2,000 drugs, it means the ratio is 50%. So more than 50, nearly 50% of the drugs are from natural origin. These are the some of the anti-cancer compounds which are plant derived, and they are in clinical studies. Even some of them have been passed through all the clinical trials, and these are resveratrol, curcumin, EGCG, quercetin, betulinic, artemisinin, rutin, ginsenosides. So resveratrol is used in liver, colon, breast cancer, neuroendocrine cancer, and this is vitis vinifera is it is grapefruit. So this compound is originated from vitis vinifera, and it has been explored for this 4 type of cancer, and it has passed clinical studies. And curcumin, it's turmeric. It is well known for curcumin, bis-dimethoxycurcumin, and then dimethoxycurcumin also, which is originated for curcuma longa, which we call it turmeric. It is a well-known spice, and it is explored for 3 different kind of cancers. And other are EGCG, which is obtained from tea. Quercetin, it is a flavonoid mainly found in many fruits and vegetables. It's common flavonoid, and it has been found in vegetables also. Then artemisinin is an anti-malarial drug. Apart from anti-malarial property, it has been explored for this breast cancer, cervical and colorectal cancer also. And this is ginsenosides, which is obtained from Panax ginseng, and there have been many compounds from this group have shown promising anti-cancer activities. So this beautiful slide, you can observe these are your phytochemicals in your day-to-day spices and other kitchen things you may find. First one is clove, which you use when your teeth aches, and then nutmeg, pericarp, then cumin. This is saffron. Saffron is well known for neuroprotective effects. The other one is we call fenugreek, methi, which is a very well-known anti-diabetic property, and this is ajwain. Then this piper nigrum, which is known as black pepper. It is piperine and piperidine. These are alkaloids which are mainly found in piper nigrum, and it has very much broad spectrum of biological activities. Then it is cardamom, then turmeric, nutmeg, then red pepper, then this is coriander. This is I think this is cardamom, and this one bigger cardamom, and this is cinnamon, then star anise. These all compounds are responsible for biological activities, as well as these are we are using these for fragrance purposes in our cuisines. So let's start the journey from collection to confirmation. This chart is very important to understand how the things happen when you do research in natural products. First step is collection of plant materials. Suppose if you are interested in a plant, then you have to collect it from field or farm or some wherever it is found, then you have to authenticate it. Authentication is very important step because there are many similar plants to each other. If you are talking about tulsi, then you might have heard about Shyam Tulsi, Ram Tulsi. So whatever you collect, you must have its documentation, and that is why there will be requirement of taxonomist or a botanist to authenticate this plant. And then cleaning is also a necessary step, because whenever you collect it, it must be clean. So then you have to go for drying, because all the freshly collected plant material you have to dry because of the moisture present inside. And this step is very important, because if you are focusing on volatile compounds like eugenol and these essential oils or something like that, or having boiling point of less than 40, then you must dry it in acid. And then you have to store it in a airtight container, so that it may not contaminated from the moisture or fungus like that. And the second step is extraction is we will discuss this later, what does the extraction means. But there are few steps we need to know. First step is selection of proper technique, then solvent to sample ratio. How you have to choose the solvent to sample ratio, or solvent or technique, it is depend upon your choice of compound. So after that, when you prepare extract, you have to dry it in a rotary evaporator, keeping the temperature low, below 40. And if you are going for isolation, then this step need to be scaled up, because in plant extract you don't know how much yield is present. So when you go for isolation, you have to keep your plant material in kgs, and you will get the yield in grams, so you can load it in column chromatography. And if you are going for only analysis like qualitative or quantitative by various chromatographic and spectroscopic technique, then you can carry out this in grams also. But if you are targeting isolation of your compound of interest, then you must have to go for in kg. And after column chromatography, if you are getting some positive response from column chromatography, or you are getting some fractions which are having pure compounds, then you purify it with different chromatographic techniques. Then when you get pure compound, then you go for spectroscopic technique, so you can elucidate the structure. Now I have taken two routine examples why we are carrying out this black tea. The left picture is of black tea, why we are carrying out it in water, and the right picture shows the uh, beautiful golden color of turmeric in milk. So if you go by terminology, the separation of medicinally active portions of plant tissues from inactive or inert part, by using suitable or selective solvents in standard extraction procedures. So it says, when you are choosing a solvent, or it must it must penetrate into the plant cells, and it remove the inactive part from the your plant, and your active portion should be come out in the media. So this this structure in this black tea cup, which is, it's a one kind of polyphenol which is mainly found in you know, tea. And when you prepare tea, you have to go for water only. And then you when you add water na, one interesting fact I remembered, this black tea or green tea have biological activities such as antioxidant or anti-cancer. You might have read in newspapers and research papers also. But when you go for milk tea, then what happens? Chemical reaction between this polyphenol and milk. There is a protein called casein, which is reacted with this polyphenol, and all the antioxidant or this biological activity is destroyed. So when you are consuming a black tea only, then you will get its benefit. But when you are drinking or consuming a milk tea, then you will not get that much benefit. So main concern was why we have selected water as a solvent here, because green tea polyphenols are, or black tea, green tea or black tea, there must be polyphenols inside. So it you know, extracts in water. That's why we choose water as extraction solvent here. And curcuminoids are fat soluble compounds, and milk is itself a fat. So when you are in a cough or cold like situation, then your ancestors will ask you to consume a uh, hot milk having this turmeric powder added. So this is the basic thing we must know. Now, all these things you can observe how this happens. Like curcuminoids are solubilized in the this milk, but your catechins like polyphenols and other compounds are solubilized in water. So it depends upon the polarity, and the universal law says like dissolves like. So when you are targeting a compound, you must know the polarity of your compound, of your the targeted compound, and for that you need to choose a solvent which are capable of extracting your compound of interest. So these are the some of the common solvents which are used in this plant material extraction. You have to choose n-hexane or petroleum ether for non-polar compounds. Then these yellow marked compounds are solvents are medium polar, and these blue ones are methanol, ethanol, water. Water is known as a universal solvent, and it can, you know, extract anthocyanins, tannins, saponins, terpenoids, etc., these compounds. But when solubility of polar compound in water is very less, then you have to opt either methanol or ethanol. And when you go uh, for a medicinal property, then you cannot use methanol as such. So you have to go for ethanol. We can say some of the homeopathic medicines are most of the homeopathic medicine tinctures are made in ethanol 95%. So either water or ethanol you can use for direct consumption, but you need to dilute it somewhat for ethanol. But you cannot consume, these are not safe. But for natural product research, when your compound solubility is lower than this in a polar solvents, then you have to opt between these solvents, whether they are extracting or not. This is illustrative drug discovery process, which takes not more than 10 years, because it's a very long process. Suppose if you have unknown plant material, then you are extracting it with different kind of solvents, as I have mentioned here. Suppose you take three solvents, N-hexane, and from medium polarity solvent you are taking ethyl acetate, and then for polar fractions you take methanol. So suppose you have unknown compound and you are targeting a compound which are soluble, which solubility of that compound we don't know, then you have to opt for different kind of solvents. This is particularly I have shown for how the drug discovery process takes place. Now, this is in small scale, and when you get positive results, then you have to go for large scale, which is I have mentioned as step 2. So first, you are taking three different solvents, and extraction is done by this Soxhlet extraction method with three different solvents. Suppose out of these three different extract, you are getting positive results with a particular in vitro assay, like anti-cancer or antiviral or anti oxidant, whatever activity you are targeting, you just do the in vitro assay of these three extract, and if you are getting positive results, then you should go for this large scale extraction, and again repeat the in vitro assay, whatever activity you are targeting, and after extraction in large scale, you are getting active extract again, then you do column chromatography, then again you perform bioassay, then you are getting also active fractions, then you are going, it means that whatever results you found in step 1, you are getting the same results up to this fractionation, then you are going in right direction. Then also again do the fractionation or isolation of pure compounds with the help of different kind of chromatographic techniques, then you get a bioactive compound. It might take it's a very tedious and not everyone's cup of tea, but you need to have enough knowledge of this spectroscopic and chromatographic techniques. Then when you get isolated compound, you determine its structure, and when you get the compound, you also perform its bioactivity. If it is as similar as you get in extract, then you are going in right direction. You can assume that the particular bioactivity is responsible for due to this isolated compound. And these are the steps after isolation and your compound structure determination. In vitro assay, then you have performed here in vitro assay, then you have to go for in vivo assay and its toxicity study, then again go for clinical trials, then patent applications. Whatever you are getting results, you patent it, and ask for approval to regulatory, and then after getting all the approvals and all the documentation process, you can go for its production in bulk. You can sell your patent or whatever you want to do, but these are the illustrative drug discovery process, and in real it seems like a very easy, but it takes a lot of time and also expertise. It takes nearly 8 to 10 years. These are the some of the extraction techniques. Whatever we are performing in lab at a small scale, we can say conventional, which do not require any kind of special equipments, but they have some sorts of disadvantages also. And there have been emergence of non-conventional techniques such as microwave ultrasound assisted or microwave assisted supercritical fluid extraction, and these are the other extraction techniques are also in existence. And it takes very less time compared to these conventional techniques. We'll discuss one by one. Out of these 6 non-conventional techniques I have mentioned here, 3 which are very widely used. One is microwave assisted extraction, then ultrasound and supercritical, which are very helpful to understand how the things takes place. First of all, maceration. Maceration is nothing, just you have to put your plant material, whatever it is, if it is leaf, fruit, stem, bark, or root, whatever sample, you have to go for its powdered material so the solvent can reach each and every place. And frequent stirring is preferred, but if occasional stirring is given, then it is okay. You have to cover this vessel, because if this solvent is volatile, then it will get evaporated, and it takes hours to weeks. So we need to close this vessel, and you have to keep until the extraction completes. So it takes at room temperature, so the temperature will not be more than 40 or like that, whatever room temperature it is. There are no chances of degradation of thermolabile compounds are which are degraded at higher temperature. These compounds are known as thermolabile compounds, but when you go for heating, then these thermolabile compounds will be escaped. There are several disadvantages of this method. This is conventional method. So whatever solvent you are taking, suppose you are taking 1 gram sample in 10 ml, then every day you have to decant the solvent, and so it takes hours to weeks, and it consumes a lot of time as well as solvent. And the compounds which are not solubilized at room temperature, you are not allowed to go for this method, because it is not feasible to extract the poorly soluble compounds at room temperature. We'll discuss whatever extraction techniques are compatible for the heat sensitive compounds which are having solubility at higher temperature. Another method is percolation. For percolation, there is a vessel, cone-like vessel is required. Here also we are soaking the plant material at room temperature in a suitable organic solvent, and you can see this brown part is your plant material. You are filling it with the organic solvent, which is also known as in terminology menstruum. There is a plate and drain valve. You have to take the filtrate in a vessel, and this also takes a lot of time to complete the extraction. When your solvent is mixed with this plant material, then you have to give time to reach the this plant material, allowed to reach the your you know your plant material is allowed to soak in this particular solvent for some time, and whatever filtrate is coming out, you have to collect it in a vessel. These are the methods which are possible for uh, a lab student who can perform easily. You just require a Soxhlet apparatus. Soxhlet apparatus is given after the name of Soxhlet named scientist. It is a very ancient method because it was it was invented in 18th century, and it is very classical method. Till date we are using this method because it's a wide applicability, as well as it is exhaustive process. You have to heat the solvent, you have to fill your plant material inside the filter paper, then your organic solvent is heated. These vapors are going to condense, and the droplets of these vapors will be fallen into this plant material, and from this siphon tube your whatever extract will be fallen into this uh, flask. So it until this solvent is there, you will get the colored colored filtrate. When extraction is completed, then you will get clear or colorless filtrate. So it is an exhaustive process, consumes lesser time than maceration and percolation. Also, solvent used in this method is lesser than those methods. The other one is steam distillation. Steam distillation is essential for, it is best suitable method for uh, essential oils like lemongrass oil, eucalyptus oil, etc. Clevenger is an apparatus name. You have to fill your whatever material with water. Then we know that water and oil are immiscible to each other. So when you heat the plant material, then vapor gets evaporated and condensed, and this with this water, your oil phase will be separated. So you can drain out the water, and you can take essential oils, and you can analyze it, whatever compound presents, with the help of different chromatographic techniques. Here the major drawback is flask is heated at the boiling point. Suppose here we have filled methanol as a solvent, so methanol will be heated up to 70-80 degrees centigrade. So there may be chances of degradation of compounds, and here we are taking water as a extraction solvent, and is heated above 100 degrees centigrade. So there are large there are chances of degradation. So these are the major disadvantages of conventional methods. As we know that in maceration or percolation, it takes weeks to hours to weeks, even 3 days it may take 5 days also. So longer extraction time is the major disadvantage. Other, efficiency of the targeted metabolites is lower. High temperature also a major cause, which leads to degradation of compound of interest. Solvent consumption is also high, so cost will be high, and the process are tedious because when you perform a maceration or maceration, percolation, or Soxhlet, whatever, you have to filter it, or you have to evaporate it large amount of solvent, and it harms your health also. and the reproducibility of these methods are very less compared to the non-conventional technique. Let us move to non-conventional technique, and these are the three techniques which will be discussing: microwave assisted, ultrasound assisted, and supercritical fluid extraction, which are the major three extraction techniques which are used. Uh, First of all, we'll discuss with microwave assisted extraction. So microwaves are nothing but electromagnetic radiation which falls in between the range frequency ranges 300 gigahertz to 300 megahertz, and there are two principles behind the heating. One is dielectric heating caused by rotation of dipole moment. So this is I have underlined because this dipole moment is required. Your solvent must have dipole moment, so there will be molecular movement, and with the friction of molecules or collisions between them will cause the heating. And the other one is ionic conductance, means friction of ions. We can easily understand this practically also. Suppose you have pure water in which ions are not present, you just uh, give it 3-5 minutes radiation, microwave radiation, and you have tap water, you give it 3 to 5 minutes of irradiation, you will get the temperature difference between them, because those who have more ions, there will be more friction and there will be more heating, and this is the major cause of heating in microwave assisted extraction. Here I have mentioned three different kind of solvents which are categorized for according to their microwave absorbing capacity. DMSO, methanol, ethanol, these are polar solvents, and they have higher dipole moment, and due to that these are heated easily as compared to the water, acetic acid, butanol, DMF, because their capacity to absorb microwaves are lesser than that of DMSO and two other alcohols. The other non-polar, such as hexane, toluene, dichloromethane, these solvents are non-polar, and hence it is also known as microwave transparent solvents. So when you go for microwave assisted extraction, you must go with these solvents, otherwise your heating will not be done because of absence of dipole, negligible or absence of dipole moment, which leads to no heating. So so this is the one of the drawback of this method. So you target whatever compound, it must be polar, or you go for other techniques for non-polar compounds. So the major advantage is reduction of extraction time. It takes only second to 30 minutes. Also, suppose you are using this method for organic synthesis, also it is helpful, because some of the organic reaction takes 30 minutes, 40 minutes, but you can do the extraction or synthesis within few minutes. And solvent requirement is also 5 to 10 folds lesser than that of conventional methods. You have large window of optimizing parameters such as extraction time, temperature, solvent concentration, and solvent to liquid ratio. Extraction time, you can you can choose different kind of time, like 3 minutes, 5 minutes, 7 minutes, like that. You can observe the yield, You can also observe the at what temperature it causes the more yield. And solvent concentration, suppose you are going with 50-50 methanol water, or you are going with 70-30 methanol water, whatever yield you are getting, you just record it. Or you also go for solid to liquid ratio. Suppose we are taking 1 gram solid and 10 ml of solvent. What happens when you take 10 gram of plant material in powdered form and you add 20 ml of water? So different ratio you can choose. And also it also gives us to change the power of the microwave irradiation. So the heating is done with microwave irradiation, so heating will be homogeneous from all the directions. Here I have taken one example. Here, see, curcumin, it is a major component from turmeric. So see, you have taken here 40 ml acetone. So acetone is comparatively less polar than methanol. So when you add, you have to if you are going for a non-polar compound like curcumin, fat soluble compound, you just have to add a little bit amount of methanol, so you can get the curcumin in the your media. And it takes only 4 minutes, which is very less. And also you can get 27% more efficient extraction yield. The another technique is ultrasound assisted extraction technique, and it uses high frequency sound waves in the range between 20 kilohertz to 2 megahertz. A normal human can hear up to 20 kilohertz. So beyond that, we use in ultrasound assisted extraction. So this is the principle how the this cavitation or whatever heating is uh, happen in this media. This is your plant. Green part is your plant matrix. Whatever you have taken, plant powder, stem, leaves, or pulp, whatever. This is your solvent. There is no barrier of solvent like microwave. You can take any solvent. And these red colors are your active part or active compounds present inside the plant material. This is the surface. So what happens when you go for ultrasound extraction? The due to the due to the cavitation, due to the sound energy, the cavitation takes place, which collides with this plant matrix, which leaches out your compound. Here I have shown. So this released molecule you can get in your filtrate. So it's a one kind of green technique because it uses very less solvent compared to the conventional method, and time also. And compatible, this is the major advantage as compared to micro assisted solvent extraction, because you are not taking any you have no barriers of choosing a solvent. You whatever you choose, polar, non-polar, moderately polar. So it is compatible with any solvents. Cost effective is also major uh, plus point of this method, because you are using less solvent and time, also manpower. And you have also, there are many optimizing parameters: extraction time, temperature, solvent concentration, solid to liquid ratio, power, frequency, whatever. You can optimize your method as per this. parameters. One example I have shown. Sorry. Okay. So polyphenols, you are extracting polyphenols from pomegranate using this method. It takes 1 hour as compared to the 72 hours in traditional maceration method. So you are saving your time, money, and everything. The other one is SFE, supercritical fluid extraction. Before going to this technique, let us understand the phase diagram. Phase diagram is important thing in this particular extraction, because whenever you heat or you are giving heat or pressure to any gas or like carbon dioxide or oxygen or water, whatever, uh, it 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 goes beyond your critical point. There is a phase, there is a region which is known as supercritical fluid region, in which it behaves like a liquid, but its power is like gas. So we are choosing carbon dioxide as a solvent for this extraction technique. And there are critical parameters for carbon dioxide. Is this critical temperature is very less, 31.1 degrees centigrade, and critical pressure, it is very less compared to water. That is why we are not using water as a solvent here, because you have to go beyond 374 and 217 atmospheric pressure to achieve uh, this homogeneous fluid which behaves like liquid and having power like gas. So other reason behind taking CO2 is non-toxic, is non-inflammability, lower critical constants, which I have here highlighted. Also, it is inexpensive, contaminant free, and you can control this with the help of the software, and safe to use because you are using very less amount of solvent. And also carbon dioxide is recyclable, so you can recycle the solvent after the extraction also. And this method is widely used for non-polar or essential oils or food grade sample, or like lipophilic compounds which are not solubilized in what polar solvents. So you can also here extract with the help of carbon dioxide. In this method, you can add ethanol or methanol, which are known as modifiers, and it should be less than 10 to 15. And these are also added because to leach out the compounds which are polar in nature. So methanol and ethanol are used as modifiers. And here are two examples: artemisinin from Artemisia annua plant, uh, and caffeine from this Theobroma cacao plant. Now, after extraction, the next step is whatever you are targeting is present or not. Suppose you are targeting a flavonoids, then you have extract and you don't know anything about the plant. So you can perform this test. These tests are qualitative. You can know the presence or absence of these phytochemical groups such as flavonoids, terpenoids, alkaloids. You can find this test in textbooks also. The one of the technique which are widely used for natural product research is HPTLC. It is nothing but modern version of TLC, thin layer chromatography, which we are using for organic synthesis also. So here in nature product, you can use this technique with densitometry for qualitative, quantitative, and even preparative purposes also, micro preparative. You can isolate your compounds with the help of HPTLC. So you have to select a stationary phase, either normal phase you are going for, or a reverse phase. Then you have to select the solvent, which is known as mobile phase, and optimize it with the different proportions. And also you have to your solubility of the samples must be in a particular solvent, so you can generate a fingerprint. And after development, you can detect the compound. And in HPTLC also, we can validate the method. For quantification, validation is necessary, and these are the some of the validation parameters which has to be carried out in validation. These are the several mobile phases of different compounds. Whatever compounds you are going for, you have to choose the mobile phase according to that. And if some compounds do not, hello, sorry, you have to go for derivatization. And for that, you can use different kind of stains. uh, okay, we'll go fast because of time constraint. Some of the papers which have been published by our research organization, these are the Giloy extract, which are there are three compounds which are major in this extract. Tinospora cordifolia, its botanical name, and it is used for immunomodulation. And this one is the HPTLC MS equipment we have here at Farmenza Herbal Private Limited. You can scrap out whatever spot you want to identify or confirm it with the help of mass. Just scrape it out, whatever spot you are targeting, and you can generate the mass spectra, and based on that you can confirm the compound. This is the another example of Berberis aristata, which is known as Darul Haldi. These are the one of the important method, HPLC, which is also used in pharma, but in natural product it has major role. And mainly UV visible and PDA detectors are used for chromophoric compounds. But if your compounds is not having chromophoric group, then you have to go for other detectors. Okay, as I have discussed earlier, in HPTLC you have to select for suitable stationary as well as mobile phase, and also validation parameters has to be carried out for quantification. And quantification is being done after validation. This parameter has to fulfill with as per the ICH guideline. These are the some of the examples which I have shown here. This is the chromatogram of 13 compounds which are found in Tulsi, and based on their polarity, it gets separated: flavonoids, phenolics, and terpenoids. It is published in Natural Products. The other example is my own research work, which I have carried out during my PhD. This is a chromatogram of 5 compounds which are present in Kokum species. These are some of the recent publications during the last few years. This is just would have possible because of our research director's vision and his willingness to do the research, and our team's hard work and commitment. Uh, Thank you. If anyone have question, please share your suggestions or questions, so I can. Am I audible? Is it okay? Your speaker is off, I think. Yeah. Hello.
Yes, sir, you are clearly audible.
Yeah.
Actually, there is no query from student side.
No problem.
Okay. Okay, so thank you once again, sir.
Yeah.
We also invite you for research collaboration with Department of Chemistry, Parul University.
Yeah.
So let us know if any collaborative research work is possible.
Yeah. No problem. We will discuss it later on. No problem.
Okay, okay. Okay, thank you, sir, for your time. Okay,
Thank
so
Thank
here
you.
I am ending this session.
Yeah.
Okay, thank you, sir.
Okay. Okay. Thank you.